is the entrance of the experimental hall where the PhDs have their own setups running. And on this wall, there are all the posters hang from the PhDs that passed through Wetsus in the recent years. From the dressing room, where we can get our lab coat, we can now enter the experimental hall. And now we enter the experimental hall, where we have more than 50 setups and where my colleagues, PhDs, but also postdocs, can run their experiment and their research. And now, some of my colleagues will explain and describe to us their own setups in the experimental hall. And the first one is Amanda, which is working on a technology to produce ultra-pure water.
Hello, I'm Amanda. I'm from Indonesia and I'm the first year postdoc at Wetsus. And currently I'm working with biological oxygen dose activated carbon with two other PhD students, Sarah and Olga. This biological oxygen dose activated carbon is using to remove micropollutants and also to producing ultra-pure water, which will be used for industrial purposes.
And now we go to the setup of Jinjian, which is working for our environment to reduce the level of CO2 in the atmosphere.
Hi, I'm Qingdian. I'm from China. And as Carla just introduced, we not only deal with water technology issues, we also try to go a bit broader to solve more environmental issues like global warming. So for my project, we're trying to use material called ion exchange resins to capture CO2 from the atmosphere. And then we combine this adsorption together with an electrochemical desorption, and we can produce high purity CO2 gas. Maybe you are the next one that can use the high purity CO2 to produce more chemicals or fuels.
And now, passing through all these setups, we get to the setup of Anthony, which is working on CDI to desalinate water.
Hello everyone, I'm Anthony. I'm from India and I'm a third year PhD at Wetsus and I'm working on a project called capacitive deionization. It's a desalination technology to remove ions from saline water, and in my project I focus to make it more specific to remove certain ions.
And on this side, we have Olga, which is working on strategies to prevent the growth of biofouling.
Hi, my name is Olga and I came to Wetsus from Poland. I am now entering the fourth year of my PhD, in which I'm investigating the influence of pipe surface on biofilm formation in drinking water distribution systems. In my project, we try to develop coatings that would prevent, indeed, bacterial adhesion and formation of such biofilms.
And now we enter our microscopy lab. And Yuja, which is working with fluorescent microscopy, will guide us into our facility.
Hi, my name is Yuja. I'm a fourth year PhD. So today I will show you our microscopy room. In this room, you could notice there are three equipments. There are two behind me and one here. The two equipment behind me, they are epifluorescent microscopy. So you could use this equipment for your cell observation. And besides me, these are confocal laser scanning. So this is the equipment that I mainly use to analyze my soil particles. So I could use this equipment to analyze the 3D structure of my soil aggregates. Together, I could visualize bacteria and other chemical compounds. Of course, this confocal microscopy doesn't only apply for soil samples or doesn't apply only for biological samples. You could use this confocal laser scanning for any other non-biological samples such as membrane or nanoparticles.
Hi, my name is Miguel Tedesco and I work at Wetsus as Scientific Project Manager. Today I'm here in the Wetsus building where all the research takes place. So behind me, you can see the analytical lab. In this lab, all the students and researchers, they prepare their samples and they are also supported by our analytical team to make all the analysis. And analysis can go to a wide spectrums from biochemical to biological to chemical analysis.
So we have visited our microscopy lab, our experimental hall, our analytical lab, but as Wetsus is a multidisciplinary research center, we also have a multi-equipped microbiology lab, which not everyone can access though. So Olga is waiting for us inside to give us an introduction of it.
Hello everyone, it's me again, Olga, and now I would like to show you our microbiology lab. So our microbiology lab consists of two main rooms. In the first one we do our basic microbiology, where we prepare media, we grow our bacteria, we work in sterile conditions. The other room is molecular biology room. In that room we are working with DNA, RNA. We have equipment such as qPCR, PCR, but also flow cytometry and additional bright field microscope. So behind me there is a third room in which I don't have access to. Only few PhDs can enter, because there we are working with GMOs and viruses.
And maybe you're wondering if I do have a setup. I don't have a proper setup, but in my project I need to synthesize nanoparticles. And together with other people that are also synthesizing membranes, we have a synthesis lab where all the synthesis can be performed. Let's go inside and take a look. And here we are in the synthesis lab, where we have all the specific material, chemicals, tools and equipment to perform synthesis of our materials from membranes to nanoparticles, like in my case. And here I can perform my wet synthesis with controlled conditions.
Now we'll continue our tour in the lab. I would like to show our technical workshop. And now I am in the technical workshop of Wetsus. This is where all the setups are built. When you join Wetsus as a PhD researcher, you don't do research alone, but you always work in a team. And especially, every PhD researcher is supported by a dedicated team of technicians. In this way, you will be able to build your setup in no time. And you can see here, for example, how this area is used for building all the setups, going from hydraulics to electronics to parts that are 3D printed. We hope to see you at Wetsus.